Okay, phylogenetics is the study of how different species are related to each other in evolutionary terms. So what I've drawn here is a small part of a phylogenetic tree. Now, we know that human beings and chimps and gorillas are all pretty related to each other. But by looking at the morphology of the different organisms, geneticists have worked out that humans and chimps are quite closely related, and they have, at some point in the past, they had this ancestor here. They shared a common ancestor. Evolution happened. Humans one branch, and the other branch evolved and eventually became chimpanzees. We're also fairly closely related to gorillas, but not as closely related because gorillas, they do share a common ancestor with humans and chimps, but it's one branch further back. So that blue ancestor there, some of them evolved to become gorillas. Another group evolved to become this red thing here, which is no longer, that species no longer exists. So a common ancestor, and some of them evolved to become humans, some to become chimps. And you can extend it further back. For example, baboons, they're even further back. So baboons, gorillas, chimps, and humans all share this black ancestor here, this common ancestor. So that's a phylogenetic tree. And in the past, these were always put together by looking at the morphology of the organism, which sometimes can lead to mistakes. Let's look at a bit of a trivial example, but it illustrates it quite well. If you think we've got birds, bats, and human beings, somebody might say, right, well, birds and bats are the most common, or the most closely related there because they've both got wings. We're all vertebrates, we've all got blood and so forth, and so human beings, we had this other common ancestor, which is more distantly related than the ones that birds and bats share. Now we know that's completely wrong because, of course, bats are mammals and they must have evolved wings separately. Birds evolved wings and bats evolved wings, but they don't share a common ancestor. The wing evolved on two separate occasions. So the correct phylogenetic tree would be humans and bats. We share a common ancestor because we're both mammals. A common ancestor there. And birds, well, we do share a common ancestor with birds. It's a lot further back. And so that will be the correct phylogenetic tree. As I say, in that example, it's quite obvious that the first way I've drawn it is incorrect. But sometimes when you're looking at small differences in morphology between humans and chimps, for example, it's not always obvious. So about 1960s, there was a new way of doing it, which is called molecular phylogenetics. Now, the way it's done now is people look at DNA sequences, but when it first started off in the 60s, DNA sequences weren't readily available, and so the first people to do it, Linus Pauling, who was the discoverer of the alpha helix, won the Nobel Prize for that, but he decided to look at the protein, the sequence of amino acids in the beta chains of hemoglobin. Okay, so, and here is some of the results. So just make that a little. So that's the original paper there, molecular timescale for human evolution. Okay, if you want to just see that, I'll make it a bit bigger. Okay, that was actually not Paulus's original paper, but one tiny bit later than that. So if you want to look that up, and let's have a look at this table, which is taken from there. Okay, so they looked, there's about 400, I think it's 417 amino acids in the beta chain of hemoglobin. And they looked at the number of amino acid differences. Okay, so every time one of the residues was replaced with another one, that was scored. Okay, so man versus chimpanzee, you can see here, are beta chains are identical to chimpanzee beta chains. There's no differences at all. Whereas if you look at gorillas, yeah, we've got two. There's two different amino acids. It's all very, very similar. Okay, then if you go back to more distant relatives, such as monkeys, so yeah, we are evolved from this. We do share a common ancestor with monkeys, but it's way further back than the ones we share with chimpanzees. You've got 12 different differences there, and so on. Now this was, and then if you look at a horse, we're not very closely related to horses at all, and it's quite a lot. There's 43 differences out of the 417 there. Okay, so this idea of a molecular clock gained currency around this time. Okay, so it's quite obvious that the more amino acid differences you've got, the further back in time our common ancestor is, the greater the evolutionary distance. And attempts were made to try and put a sort of number on this. Like, I don't know what the exact was, but it was one amino acid difference per million years. It's actually not far off that. That is falling into a bit of difficulty, really. That the whole idea that you can be so exact, and the different ways of calculating the molecular clock gives very different answers, and so it is a little bit controversial. But nevertheless, the general principle is the same. The more differences in the amino acids, that means there's more differences in the DNA. You've had more opportunities for mutations to occur, so it must be a longer time since the two species diverged from each other. Okay. Now, I think using this kind of approach, the molecular clock approach, and I think there's general agreement with this. So we just have a look at so we've got humans and chimpanzees and gorillas. Okay. Right, that's not to scale, but I think the current clever money is on this being about 5 million years. So between the time from our common ancestor with the chimp was about 5 million years ago, whereas this distance here between the common ancestor of the chimp and the common ancestor of the gorilla is quite a bit small. It's about 0.3 million years. Right, that's the current idea. Now these numbers have come not from looking at hemoglobin. They've come from looking at DNA sequences. And the ones that they used to do this were the mitochondrial DNA. And mitochondrial DNA is like prokaryotic DNA. It is found, the mitochondria were probably a prokaryotic organism which became lives in a symbiotic relationship with a eukaryotic cell. And so they don't have eukaryotic DNA with chromosomes and so on. They have a single circular chromosome. It's pretty small, and it codes mostly for proteins which are found in the mitochondria itself. So it's it's a bit easier to work with than the whole of the human genome, because it's one small, relatively small section, and a lot of studies have been done on that. Comparisons of the DNA sequences of humans, chimps, and gorillas and other primates, and this current time scale has been suggested from that. Okay, now just let's have a look at another way of doing molecular phylogenetics. Is looking at the DNA sequences of ribosomal RNA. Okay. And there. Ribosomal RNA. Now, ribosomal RNA is The ribosomes are made up of proteins and of RNA, and there are genes for those ribosomal RNAs in the human genome and all animal genomes, and they've been compared with each other quite extensively for phylogenetic reasons. So let's have a look at some results from that. Okay, so just get this figure here. Right. Okay, this is not very easy to read, but what this hue there, this is human. This is a small section of ribosomal DNA. These are the numbers of the bases. So that's 304. That's down to 241. The numbers here. This is human. This column here is HU. That's human DNA. This one here, the next one down is chimpanzee. The GO, that's gorilla. OR is orangutan, which are primates. And then finally we've got mouse, M, and we've got rat. So they're a lot more distant to us. Okay, so these gaps here, that's where they haven't put the sequencing because they're not of interest. And the numbers up here, they put a number where you get marked difference in a base sequence between the different primates. So you can see that one, number 29 there. It's C in humans, C in chimpanzees, it's T in gorillas, it's C in orangutans, and so on. So you can see there are a lot of areas where the ribosomal RNA, the sequences match up very closely, but there's also areas clearly where there have been mutations. And by looking at those mutations, and it's a very laborious procedure, it's obviously got to be done by computer because there's vast numbers of bases in the sequence. These phylogenetic trees can be worked out. Right. Topically, I've got another phylogenetic tree here, which looks extremely complicated. But if you look at that red one there, you can see it in red. It says WH human 1 China 29 December. That is the coronavirus. That's CoV SARS-2. That's the virus which causes COVID-19. And this shows that it's actually very closely related to this. So you can see the SARS-CoV there, that one there. That is the virus which caused SARS in 2013. Sorry, in 2003. That was an outbreak which killed a lot of people in the Far East, in China and in Singapore. And there was also a small outbreak in Canada, but it never really became a pandemic in the same way as the CoV SARS-2 has. So that just shows the evolutionary interaction of those things. You can see that they're coming from, they share a common ancestor, and all the other things there, which there's hundreds and hundreds of them. Well, not hundreds, but there's a lot of them there. There's another one there called MERS, which I've just spotted. Okay, that causes respiratory distress, like the CoV SARS-1 and CoV SARS-2, and that was Middle Eastern respiratory in the Middle East, and it jumped species from camels. I think that was about 2007 or something that happened. So they're all fairly related. And you can see there are a whole load of other viruses there which have been isolated from bats mostly, I think, all coronaviruses, and they've all been sequenced and characterized, and that is a phylogenetic tree of those, which is of obviously contemporary importance.